In this video, we will go over the de novo biosynthesis of purines. The two main purines found in the body are adenine and guanine. So it's important we recognize that our body is undergoing a de novo biosynthesis. De novo basically means from scratch. So it's going to pick up and build these two nitrogenous bases, which will later be incorporated into DNA and RNA. It will do so from scrap metabolites found in the body. So shown here is the skeletal structure of a typical purine. This is not the structure of adenine, nor is this the structure of guanine. This is the basic skeletal structure. First, let's see where the atoms come from. Carbons 4, 5, and 7 come from the amino acid glycine. Carbon number 8 comes from formate. Same with carbon number 2. Notice that purines are a bicyclic structure. They have two rings. The amino acid aspartate donates nitrogen number 1. CO2 or bicarbonate gives rise to carbon number 6 in the purine backbone structure. And finally, the amino acid glutamine, which is known to be a major nitrogen or amino donor in the cell. Glutamine here donates its nitrogen atoms. So here we have amino acids, CO2 or bicarbonate, which is typically coming from respiration. We also have formate, which comes from tetrahydrofolate. Tetrahydrofolate is a carbon carrier, and it's going to carry the formate and carbon's oxidation state of formate. It will carry that and donate it as formate, those carbons as formate, to carbons number 2 and carbons number 8. So you can basically see how we're sort of building from scratch, which is essentially what de novo biosynthesis is. Important when discriminating the differences between purines and pyrimidines is that purines are actually made with ribose 5-phosphate already attached. So ribose 5-phosphate comes from the pentose phosphate pathway, and ultimately it's made from a molecule known as PRP. So ribose 5-phosphate generates PRPP, phosphoribosyl pyrophosphate, and then it's on. This skeletal block of purines are built on the ribose 5-phosphate backbone. A fundamental difference between this and pyrimidines, cytosine, uracil, and thymine, is that the ribose 5-phosphate, that sugar and phosphate moiety, are added in the very last step in pyrimidine de novo biosynthesis. So before we embark on the 11-step pathway to make by de novo synthesis the purines, adenine, and guanine, the cell first makes this molecule here. This molecule is known as inosine monophosphate. A couple of things I want to bring your attention here is that this is monophosphate. So there's one phosphate attached. And then what makes IMP, inosine monophosphate, actually IMP, is the nitrogenous base. So the nitrogenous base here is hypoxanthine. So this is what we make first in the 11-step pathway. We build this on the ribose 5-phosphate backbone, and then we have a branch. The IMP will bifurcate and make in a 2-step pathway either AMP or separately in a separate pathway, GMP. So IMP represents the branch point for de novo purine biosynthesis. From IMP, the cell has to make adenine in the monophosphorylated form or guanine in the monophosphorylated form. Again, the ribose comes from ribose 5-phosphate. We build this on the ribose 5-phosphate scaffold. So here is our 11-step pathway. Notice a lot of these enzymes are multimeric, which means that they're subject to high levels of regulation and perhaps allosteric regulation as well. In fact, this is very much a regulated process. It's important that the cell maintains a balance of AMP, GMP purines, as well as the pyrimidine, cytosine, uracil, and thymine. Uracil is found in RNA, Thymine is found exclusively in DNA. And here we make IMP. Again, to reiterate the point, it's made on the ribose 5-phosphate backbone. As we can see here, we're adding atoms by atoms. The nitrogenous bases are added to the ribose 5-phosphate. The ribose 5-phosphate comes from the pentose phosphate pathway, and we prime the ribose 5-phosphate. The way we prime it is by making this molecule here, PRPP. So it's really built upon PRPP. The pyrophosphate leaves, shown here. Once the pyrophosphate leaves, it's on to the races, and we just build on the sugar and phosphate moiety. So, as illustrated before in the previous slide, ribose 5-phosphate, ultimately coming from the pentose phosphate pathway, is utilized to generate this targeted, activated, or I should say primed molecule. And the primed molecule is PRPP, 5-phosphoribosyl alpha pyrophosphate. So the PRPP here stands for the pyrophosphate. That's a very good leaving group, and it is this leaving group that's going to allow the reaction to occur, the step-by-step synthesis on this backbone. The backbone being the sugar and the phosphate moiety. So the enzyme that does this is ribose 5-phosphate pyrophosphokinase. We will see here in a lot of this 11-step pathway, we will use a tremendous amount of ATPs. It's a very ATP-dependent process. Step 2 of this 11-step pathway, here's that PRPP one more time. The pyrophosphate is going to leave, so if we were to sort of diagram where the atoms are going, the pyrophosphate leaves. This provides enough energy to actually drive this reaction in the forward direction. Glutamine donates its amino group, and also we have an inversion of configuration. So the alpha anomer gets converted to the beta anomer, and in doing so, the NH2 is added. So the NH2 here comes from glutamine. This is reaction 2 of the de novo biosynthesis of purines. The first purine we make is IMP, which then branches out. So here is NH2, which comes from glutamine. Reaction 3 involves glycine. So this is nitrogen number 9 of the purine ring, and then glycine through this enzyme GAR synthetase is going to take PRA, beta 5-phosphoribosylamine, and convert it to GAR, glycinamide ribonucleotide, and here are the carbon atoms. It's basically this carbon atom, and this nitrogen atom that will come from glycine. As we step by step make this, again, on the backbone of ribose 5-phosphate. This came from PRPP, which sort of was the prime. It primed the initial step, and if you remember, the initial step was having glutamine add across this number 1 carbon of the ribose sugar. Here we go with reaction 4 of the pathway. Remember, this nitrogen came from glutamine, and these red carbons came from the amino acid glycine. So here comes tetrahydrofolate, donating the carbons in the formal oxidation state. And basically, this is carbon in the form of the aldehyde, and we add a CH double bond O. Okay, so the oxygen is going to eventually leave. This CH comes from tetrahydrofolate carrying carbon in the formal oxidation state. So the enzyme that does this is GAR transformylase, and here we have this carbon that's coming from tetrahydrofolate, this NH coming from the amino acid glutamine, and then once again this nitrogen coming from the amino acid glycine. We build, once again, it's very important when we want to discriminate between purine and pyrimidine biosynthesis, we build this heterocyclic ring on the sugar phosphate moiety. Reaction 5 now in our 11-step pathway requires the energy in the form of ATP. So once again, the common theme here is that this 11-step pathway is a heavily ATP dependent process. So energy is very much required to make the purines. And here glutamine is going to donate its amino group. The energy of ATP is utilized, so phosphorylasis occurs. And glutamine, by donating its NH group, becomes glutamate. And then we sort of add to the number 4 carbon here, this NH. So once again, the ribose 5-phosphate coming from PRPP stays the same. PRPP primes that initial NH addition, which comes from glutamine. This carbon, and nitrogen comes from glycine. This carbon comes from tetrahydrofolate, carrying carbon in the formal oxidation state. And then this nitrogen shown here, coming from reaction 5, is donated by glutamine, utilizing the energy of ATP. The enzyme that does this is F-gam synthetase. Reaction 6 requires no addition of any molecules. This is basically a cyclization step. So utilizing, once again, the energy of ATP. So that's about 3 or 4 ATPs that, by my estimation, are being utilized to build this ring structure. But essentially, we form a ring here. And there you see the ring structure forming, and it's a cyclization reaction. So that's reaction 6 of the 11-step pathway. Reaction 7 requires the utilization of bicarbonate. Usually that bicarbonate comes from CO2. CO2 is an active product in many types of respirations, and that CO2 can easily pick up a proton. Carbonic anhydrase does that, is the enzyme that does that. It could easily pick up the proton and become bicarbonate. So in another ATP-dependent process, ATP is utilized to sort of carboxylate the number 4 carbon. Again, ribose 5-phosphate comes along for the ride, coming from PRPP, and we're sort of building this step by step. The enzyme that does this is air carboxylase. The enzyme itself has a number of different subunits. Here you can see all the different subunits and a tremendous amount of symmetry in this enzyme. Once again, we're going to follow the atoms and where they are coming from and their origin. This nitrogen comes from glutamine. Then we added carbons 4, 5, and nitrogen number 7, which comes from glycine. Then we added carbon number 8, which comes from formate, delivered by tetrahydrofolate. Then we added this nitrogen, which comes from the amino acid glutamine. And then finally, this carboxylation comes from CO2, or bicarbonate. Reaction number 8 again requires another ATP molecule. Again, notice the heavy expenditure of the cell utilizing ATP. And this utilizes the amino acid aspartate, utilizing ATP hydrolysis to sort of attach this aspartate amino acid to the carbon here. This carbon, remember, came from CO2. Ribose 5-phosphate comes along for the ride. Now this aspartate is an amino acid. It could come from the diet. It could also come from oxaloacetate through transamination. And that aspartate also comes from the urea cycle as a sort of a way to sort of carry extra amino groups. So once again, the enzyme that does this is cyclization synthetase, utilizing ATP hydrolysis. And going over the atoms and where they're coming from one more time, this nitrogen comes from glutamine. Remember, we utilized PRPP to prime that to allow that glutamine to get in there. Pyrophosphate is the leaving group for that reaction. So the ribose 5-phosphate is the backbone. Carbon 5, carbon 4, and nitrogen come from glycine. Then this carbon came from tetrahydrofolate. We added this nitrogen from glutamine. This C double bond O came from bicarbonate, and finally in the green we have aspartate. Now the next step in the pathway, this aspartate will actually sort of cleave, leaving this NH group as part of the nitrogenous base and liberating fumarate. In fact, that is the next step of the reaction, the generation of fumarate. So reaction 9 in this pathway, this NH2 amino group came from aspartate. Fumarate leaves. We saw fumarate as a metabolite in the TCA cycle. We also saw fumarate as one of the products in the urea cycle. And fumarate, by the way, is also a byproduct of the amino acid pathway, degradation pathway of phenylalanine. So fumarate is seen a lot in different metabolic perspectives. So ribose 5-phosphate once again comes along for the ride. This nitrogen comes from glutamine. Remember, PRPP primes that nitrogen input. Then carbons 4, 5, and this nitrogen comes from glycine. This comes from formate, delivered via tetrahydrofolate. This amino group comes from glutamine, a major donor of nitrogen groups. This C double bond O comes from bicarbonate or CO2. And finally, by the leaving of fumarate, this NH2 in the green ultimately came from aspartate, the amino acid. The second to last step in generation of IMP is an addition of the formyl group one more time. So this is carbon in the oxidation state of an aldehyde. It's being carried by folate, or in this case tetrahydrofolate. The formyl group is attached to nitrogen number 10. And here we see it being attached, this C double bond OH, the aldehyde group, or also known as the formyl group, carbon in the formyl oxidation state. There's that carbon. And it's attached to nitrogen number 3. And this is the penultimate step of the generation of IMP. Once again, ribose 5-phosphate comes along for the ride. PRPP primes the reaction to add this nitrogen that came from glutamine. Then these carbons and that nitrogen came from glycine. This CH came from tetrahydrofolate. Then this NH came from glutamine. Normal respiration and metabolism gives us CO2 and bicarbonate. That's where this carbon came from. And this NH2 came from aspartate. Finally, tetrahydrofolate donates this carbon here. And in the 11th and final step, in the synthesis of the purine backbone structure, we have a cyclization reaction. The final step in generation of IMP, or inosine monophosphate, is a cyclization. So this is essentially a dehydration reaction. Water leaves, so one of the oxygens and two of the waters sort of leave as the water molecule, and you have the generation of this ring structure right here. So we have IMP, this enzyme that makes this is IMP cyclohydrolase. And you can notice here the enzyme is a dimer. Actually, I see some symmetry here, so it looks like it has some threefold symmetry. Very multimeric, essentially all of these enzymes are very much multimeric enzymes, which means they must be subject to a tremendous amount of regulation. So we are finished. We have generated the molecule IMP, which is the branch point. So once again, this NH2 came from aspartate. Remember, fumarate left to generate that NH2. This nitrogen came from glutamine. The carbon, and nitrogen came from glycine. This carbon and this carbon came from tetrahydrofolate. And this nitrogen came from glutamine, and this C double bond O came from CO2 and bicarbonate. Once we form the ring structure here, we've generated hypoxanthine. Hypoxanthine attached to the ribose and phosphate gives you IMP, inosine monophosphate. Now it's decision time for the cell. IMP is made, and we need to generate AMP and GMP, which are both of our purines. And so notice here, before we step any further, notice that to make AMP, we need the energy of GTP. Where does GTP come from? GTP comes from GMP. It's very easy to phosphorylate GMP to GDP and also GTP as well. So those are kinases that do that. Also want to reinforce that in order to make GMP, let's highlight that in green here, we need the energy in the form of ATP. Well, where does ATP come from? ATP comes from AMP. Remember, AMP can be phosphorylated to ADP, which in turn can be phosphorylated to ATP. So we have this sort of cross-regulation going on here, such that we want equal amounts of both AMP and GMP nucleotides to be generated in the cell. In fact, in a disequilibrium, or having one purine over the other, is very, very bad, because that could lead to DNA errors. So let's first go through the pathway in making AMP. Very important that it's recognized that we're making the monophosphate. And it's also very important that we're making, if this is going to be nucleic acid, we're making RNA. So you see, if we want to make DNA, we have to deoxyribonucleic acid, we have to remove an oxygen. So we're making the RNA ribonucleic acid, not the deoxyribonucleic acid. So it's important to recognize that we're making RNA first. To make DNA, we have to do a ribonucleotide reductase reaction. That enzyme, a heavily regulated enzyme, actually strips away one of the oxygens. It strips away the oxygen at the number 2 carbon of the ribose. So we're making RNA, we're making it in the monophosphorylated form of the nucleotide. So IMP in this first step utilizes aspartate and the energy of GTP. So here's our aspartate molecule. And by now we should know that aspartate can easily liberate fumarate. And we have here, let's highlight this here. Here is the NH2, which came from aspartate. So NH2 is the origin of that aspartate. And now we have AMP. So that's the difference between AMP and IMP, the amidation, or the generation of the NH2 group, which comes from or is donated from aspartate. Finally, let's see if the cell wants to make GMP. So in order to make GMP, we have an oxidation reduction reaction where NAD plus gets reduced. In turn, we have the oxidation here of this carbon. This is called xanthocene monophosphate. So we will come across this molecule later when we talk about nucleotide degradation. And then finally, to go from xanthocene monophosphate XMP to our final destination, our final purine GMP, we have to donate an amino group. The amino acid that donates that amino group is glutamine. So here we have that NH2 group. The difference between GMP and IMP is that NH2 group attached to this carbon right here. So notice the cross-regulation, notice the interdependency. Once again, to make AMP, you need the energy in the form of GTP. Once again, to make GMP, you need energy in the form of ATP. So these pathways are mutually dependent upon one another. Just want to mention that clinical relevance, there is a drug out there called mycophenolic acid, and this drug actually is an inhibitor of IMP dehydrogenase. So this enzyme, if we go back to the previous PowerPoint slide, this enzyme generates XMP from IMP. So IMP, inosine monophosphate, gets converted to XMP, xanthocene monophosphate. In that, we generate an NADH. We oxidize IMP to get XMP. IMP dehydrogenase is the enzyme that does this reaction, redox reaction. It's upregulated in certain types of cancers, and it's a target for a lot of anti-cancer drugs. And this drug is a natural product from fungus that is a target of this enzyme, inhibits this enzyme, the idea being that you can halt purine de novo biosynthesis in cancer cells by targeting this enzyme. Mycophenolic acid is one of the drugs that's out there that does this. So finally, let's talk about the heavy amount of regulation that's important to maintain equal amounts of ATP and GTP. So remember, the branch point here is IMP. inosine monophosphate, and we make XMP here. Remember, IMP dehydrogenase is the enzyme that does that. And then from XMP, glutamine donates its amino group to give you GMP. So notice immediately that GMP feedback inhibits its own generation. That's called feedback inhibition. Let's go to this side of the branch here. Going to this side of the branch, we will see that IMP gets converted to adenylosuccinate. Aspartate donates its amino group, and that's how we generate AMP. And notice that AMP feedback inhibits its own production. So we have feedback inhibition at the monophosphorylated form of the nucleotide. Notice here the diphosphorylated form of the nucleotide feedback inhibits very, very upstream here, but the part where we activate PRPP, and this is the molecule that adds the glutamine, adds that nitrogen, if you remember, in the pathway. This is reaction number 3 in the pathway. So ADP and GDP inhibit that step where PRPP gets activated through the glutamine molecule. And by the way, PRPP, phosphoribosyl pyrophosphate, feed forward activates itself. This is called feed forward activation. So the diphosphorylated form feedback inhibits all the way up on the pathway. The diphosphorylated form also feedback inhibits way upstream here at this step here where ribose 5-phosphate gets converted to PRPP. So that's way upstream in the pathway. The diphosphorylated nucleotides ADP and GDP inhibit. So there's a lot of regulation. Okay, the immediate regulation is the feedback inhibition. AMP halts its own production. GMP halts its own production. Recognize that AMP requires GTP hydrolysis and GMP requires ATP hydrolysis in order for them to properly get made. And ultimately, we get ATP and GTP in just the right amounts, should be equal in order for the purines to be incorporated not only into DNA or RNA, but also for normal cellular processes, correct? Because ATP is an energy source in the cell, GTP is an energy source in the cell. So it's not just DNA incorporation, also energy. We need to have equal amounts of ATP and GTP.